cleavage. And step 6, oxidation phase. Oxidation. What is oxidized? Glycerol 3 phosphate is oxidized into 1,3 bis phosphoglycerate, but non-plus is reduced. Therefore, we have this energy. The energy reduced coenzymes are energy as far as the process is aerobic. Okay. Now, the second product is here. Look how this molecule is generating ATP. ADP enters and ATP is produced now. It is starting to give us ATP. ATP is output here. And therefore, we are losing the phosphate at carbon number 1. So that only 3 phosphoglycerate is produced. And step 8, this enzyme is catalyzed by the same kinase. Of course, it is known as 1,3 bis phosphoglycerate kinase. Named. You name the substrate and you add the term kinase, because enzymes named by this name are catalyzing transfer phosphate groups from one molecule to, in this case, from this molecule into this, into the ADP. ADP, so that ADP gets one phosphate, becomes ATP, that is one energy. From one glycerol 3 phosphate, one ATP, from two ATPs are generated here. Because from one glucose we obtain one this and one this. One this we convert into one this, therefore we do have 2. We have to multiply everything hereafter by 2. Then what happens? This enzyme is kinase 1,3 bis phosphoglycerate kinase. Then reversible reaction happens here. That reaction is mutation to phosphoglycerate. And the enzyme is not that much important because it picks the phosphate from carbon number 3 and puts it in carbon number 2 mutations. Therefore, this enzyme is known as 3-phosphoglycerate mutase. The substrate, you name the substrate, and then you add the term mutase. And what is obtained? 2-phosphoglycerate. Now, nothing happened here. The phosphate at 3, the third carbon is put in the second carbon. And then the next step is also a reversible step. 2-phosphor, this is step 7, step 8, step 9. 2-phosphoglycerate becomes highly energetic molecule known as phosphoenol pyruvate, PEP. Phosphoenol pyruvate. And the enzyme itself is named by this term, a little bit changed to the name of enzyme, that's enolase. It's very smart enzyme. Though it is not highly controlled because it is catalyzing reversible reactions. But those enzymes which are catalyzing reversible reactions are not controlled in biochemical pathways. Now we are coming to look irreversible step. Remember the irreversible step step 1, step 3, and of course this is not a reversible step. It's reversible, of course. This one, phosphoenol pyruvate, changes into pyruvate. Let me write the formula. Pyruvate. This is the molecule known as pyruvate. This is rather the irreversible step. Sorry for my mistake. This is not reversible, irreversible. This is reversible step. The other reversible is 9, 10. The other irreversible step is step 10. Therefore, step 1, reversible, step 3, reversible, step 10. These are the steps in glycolytic pathway with irreversible reactions, so that the key enzymes which are under control, high traffic control, are these highly regulated. Therefore, enzymes here, enzymes catalyzing these steps are highly controlled or regulated. Then let's see what happens here. This is high energy molecule. This is high energy molecule. Phosphorylpyruvate. Therefore, high energy molecule is converted by giving its high energy phosphate to ADP, converted into ATP. The second payoff step. Exactly speaking, we can say that step 7 to step 10, it's payoff phase, the payoff stage. Specifically, ATP is paid off in the pathway at step 7 and step 10. From 1 glycerol 3 phosphate, 1 ATP, from 2 ATPs. Since 1 glucose results in the production of 2 glyceraldehyde 3 phosphate, we have 2 ATPs here. The same is true. 2 ATPs here. A total production of 4 ATP molecules are produced here from 1 glucose molecule, so that the net ATP produced will be 2 ATPs, because earlier, step 1 to 3, we have invested 2 ATP molecules from the cell. We have to pay that to obtain the net product, 4 minus 2 ATP produced net product. Of course, as far as the energy in glucose is concerned, you know, glucose contains a total of 288 kilojoule of energy in its form. 2,880 kilojoules of energy per glucose molecule. 1 mole of glucose contains. If we think about glycolysis net production is 2 ATP molecules. 2 ATP molecules. What is the energy in 1 ATP molecule? Almost around 30.6. 1 ATP. 1 ATP has 30.6 kilojoule. Therefore, multiply it. That is 2 times 2, 61.20 kilojoules. You see? Only 61.20 kilojoules of energy has been extracted from glucose, which contains 2,880 kilojoules of energy. This makes glycolysis highly inefficient. If you calculate the efficiency of glucose, what is the output? The output is 61. Efficiency output over input times 100. The output is 61.20, the energy that glycolysis obtained from 1 mole of glucose. The input means the total energy in 1 glucose molecule, 2880. 2880. Multiply this by 100. This, if you calculate this figure is not big. It's between 2, so 2 point something 4 or 2. That's almost 2.2%, which this efficiency makes glycolysis highly inefficient, really highly inefficient. But it's an important pathway, a starting pathway, because it has evolved during the time in the ancient planet, anaerobic environment. Still we use anaerobic respiration, that means glycolysis, to generate energy, especially in our muscle cells. Our muscle cells use, our muscle cells, in case of deficiency of oxygen, use glycolysis to generate energy. And also always there are special cells in your body which always use glycolysis to generate ATP. And these cells are without mitochondria. Of course, they faithfully transport the important molecule for aerobic respiration, that is oxygen. They transport oxygen because they have special molecule, protein, iron containing protein, that is hemoglobin. They faithfully transport oxygen from the lungs to the body cells, but they don't use it because they don't have mitochondria. So they are always dependent on glycolysis to generate